Now we shall try to understand the dihybrid cross. So let us understand the definition part of dihybrid cross. Dihybrid cross is one in which the parents involved in the process of hybridization differs in two pairs of contrasting characters or two pairs of alleles. Here, as we are taking two pairs of contrasting characters or two pairs of alleles, therefore, we are considering two characters which, let us assume, one of them as the shape of the seed and the other character as color of the seed. Now, if we look into these two characters carefully, these two characters have two variants each. So, let us take the shape of the seed and see its variants. The variants that we see, as far as the shape of the seed is concerned, the shape of the seed can be round, or the shape of the seed can be wrinkled. Now, if we consider the next character, then here also we find two variants. The color of the seed has two variants. One is yellow, and the other variant that we'll consider is green. Now, if we consider round shape of the seed to be controlled by an allele, capital R, which is a dominant allele, then the wrinkled shape of the seed will be controlled by small r, which is a recessive allele. Likewise, for controlling the color of the seed, yellow color is being controlled by an allele capital Y, which is once again a dominant allele, and the green color of the seed is controlled by small y, which is a recessive allele. So from the definition, now we can conclude that the two parents that are involved in this hybridization process is differing in two pairs of contrasting characters. So the first pair of contrasting characters are round shape and wrinkled shape of the seed controlled by the allele capital R and small r. And the second pair of alleles are capital Y and small y, which is controlling yellow color of the seed and green color of the seed. So altogether, if we consider the parents that are involved in dihybrid cross, the parents are differing in two pairs of contrasting characters, which can simply be shown as in a cross capital R, capital R, capital Y, combination of alleles controlling the specific kind of characters for this parent, and the other parent is small r, small y, controlling specific kind of characters. So these two are the parents that we are considering in this definition part of dihybrid cross. So if this is considered as a male parent, then this is a female parent. And the two parents that are involved in the hybridization technique is differing in two pairs of contrasting characters or two pairs of alleles. So now let us get into a little more detail regarding the dihybrid cross. So we are considering two parents. One is dominant regarding the two pairs of alleles that it carries for controlling specific characters, and the other parent is recessive for both the characters controlling alleles. Now, these two parents will produce gametes, and the gametes are as follows. This parent is going to produce gamete of this type, that is capital R and capital Y. Since gamete are haploid, we are representing each pair of allele only once. Likewise, this parent will produce a gamete of this kind. The gametes unite and they produce F1 generation. All the F1 generation or the progenies in the F1 will be a hybrid. That is capital R, capital Y and small y as far as the genotype is concerned. So this hybrid will express its character, and this hybrid will be round seeded with yellow color. In this case, similar to the monohybrid cross, only the alleles which are dominant is expressing themselves in the form of the character that they are supposed to express. So once again, here in dihybrid cross also, we come with the understanding of law of dominance. Now what Mendel did with this F1 hybrid is he selfed the F1 hybrid. So what he did, he took the F1 hybrid as parents 2. He took the F1 hybrids as parent 2. So the parents 2 now will have the same kind of genotype, that is capital R, capital Y and small y. And since we are considering self-fertilization or selfing of the F1 generation, the other parent that is considered is also of the same genotype, that is capital R, small r, and capital Y, small y. Now these parents once again will produce gametes and in their turn will produce their own progenies. So let us see the kinds of gametes these parents will produce. Now, to understand what kind of gametes these parents are going to produce, we shall use a Punnett square once again to find the kind of gametes or the gametic combination the parents of the second generation will produce. So let us consider the pair of alleles. The pair of alleles, capital R and small r on one hand side, and the other pair of alleles, capital Y and small y on the other side of the Punnett square. So when we combine, we see the gametic combination will be capital R, capital Y as one type of gamete, small r, capital Y as another type of gamete, capital R and small y another type of gamete, and small r and small y as another type of gamete. So these are the four types of gametes that is produced from these combination of parents. So the gametes now would be simply written as capital R, capital Y as one type of gamete, capital R, small y as another type of gamete, small r, capital Y as another type of gamete, and small r, small y as another type of gamete. Since both the parents are having the same genotypic makeup, this parent will also produce the same type of gametes. That is capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Now what Mendel did was he tried to know what will be the type of F2 generations if these are the type of gametes being produced by P2. So once again, we need to use the Punnett square in order to establish the genotypes of the F2 generation. By now we know the type of gametes that is produced by the P2 generation. So irrespective of male and female, the type of gametes produced by the second generation parents are capital R, capital Y, capital R, small y, small r, capital Y, small r, and small y. So this side we have written the female gametes. Now this side we shall write the male gamete as well. So the male gamete also will be of the same type because both the parents that was involved is having the same type of genotype. Therefore, capital R, capital Y, capital R, small y, small r, capital Y, and small r and small y will be the type of gametes for the male parent too. Now we combine the gametes in order to see the genotype of the F2 generations. So if we combine the gametes, then the genotypic combination for each individual of the F2 generation will be as follows. So number one, we combine these two, we get this kind of genotype. Another one, capital R, capital Y and small y. Likewise, capital R, small r, capital Y, capital Y. Next, capital R, small r, capital Y and small y. Likewise, we will follow similarly. Likewise, and the last one, small r, and small y and small y. So with the help of this Punnett square, we have ultimately come to the combination of the alleles from the two types of gametes, that is the male gamete and the female gamete, which is giving us 16 genotypic combination, and this 16 genotypic combination is actually nothing but the genotypic combination of the F2 generation. Therefore, this Punnett square shows F2 generations. Now, from this Punnett square, which shows 16 combination of the male and female gametes with reference to the allelic combination, we shall try to understand what will be the type of phenotypes that will be shown from these 16 combination of genotype. So let us see. Number one, if we consider this combination of alleles, it expresses a phenotype which is round and yellow. That is, this phenotype shows round and yellow seed. Now let us count the number of such individuals who expresses the character round seed, which is yellow in color. So number one, this combination will give an expression round and yellow. Here also we see both the alleles controlling round shape of the seed is dominant. One of the allele in the other allelic combination is dominant, which controls the color yellow. So therefore, this combination is also round and yellow. Coming to the next combination of alleles, round, yellow. So this one will also become round and yellow. Likewise, here also there is a dominant gene for controlling the shape of the seed, and here also in this combination there is a dominant gene which controls the dominant color. This is also round and yellow. Likewise, this is also round and yellow. This one is also round and yellow. This one is also round and yellow. This one also becomes round and yellow. And this one is also round and yellow. So if we take the total number of individuals who expresses the character, that is round shape of the seed and yellow color of the seed, out of the 16 number of individuals that is produced in the F2 generation, we see 9 out of 16 are having round and yellow seed. Coming to the next phenotypic expression. So let us consider this one. This having double R, which is capital, represents round shape, and the two small y represents or expresses the green seed. So this one becomes round and green seeded. So the next, round seed and green color seed. Round seed and green color seed. So how many of such kind of individuals are there who is expressing round and green seed out of these 16 individuals? Let's count them. So this is round and green seed. Here also we see this is round and green seed expressing genotype. So number 2. Now let us come to the other combinations and try to find out the same kind of expression, that is round and green seeded. So this is not round and green, this is not round and green. So here once again we come to find that this combination also gives round and green seed. So out of 16 individuals, 3 of the individuals are round and green as far as their phenotypes are concerned. Next, we come to this one. Now, this combination shows there are 2 recessive genes for controlling the shape of the seed and 2 dominant genes for controlling the character, which is the color of the seed. So if we consider this genotype, the expression of this genotype will be wrinkled, seeded, and yellow color. So the next combination, wrinkled and yellow. So let us see how many of the individuals are expressing this character or phenotype. So this one is expressing wrinkled and yellow seed. This also expresses wrinkled and yellow seed, and this also expresses wrinkled and yellow seed. So out of the 16 genotypic combination that is shown out here, 3 out of this 16 once again is of what phenotypic expression? Wrinkled seed, which is yellow in color. Now, the only genotype that is left out in this Punnett square is small r, small y, small y. Now, what will this genotypic expression express in terms of characters? So this genotypic expression will express this combination, as we all know, is expressing wrinkled seed, and this combination, which is small y and small y, is expressing the color green. So out of the 16 combinations shown in this Punnett square, only one of them expresses wrinkled seed with green color. So here we come to understand, out of the 16 combinations, as far as the expression of the characteristics is concerned, these genotypic combinations will show 9 individuals out of the 16 individuals are round and yellow. 3 individuals out of the 16 individuals are round and green. 3 other individuals are wrinkled and yellow, and 1 individual is wrinkled and green. This ratio is called phenotypic ratio, and this phenotypic ratio in the dihybrid cross is 9:3:3:1. That is, 9 individuals are round and yellow, 3 individuals are round and green, 3 individuals are wrinkled and yellow, and the last individual is wrinkled and green. Hence, Mendel concluded that in the F2 generation, the combination of the alleles from the parents takes place in such a manner that one pair of alleles controlling a particular character have the independence to combine with the other pair of alleles controlling other character, which he concluded as law of independent assortment. So law of independent assortment was finally concluded by understanding the dihybrid cross by Johann Gregor Mendel. So now let us try to understand the law of independent assortment. According to this law, it states that when two pairs of contrasting character controlling factors or genes are brought together in the F1 hybrid, then the pair of factors controlling a character assort independent of the other pair of factors controlling the other character. Now let us try to understand what the statements actually mean. When we were dealing with the dihybrid cross, we came across two characters which was simultaneously taken from the parents to produce a hybrid. And these two characters were, number one, shape of the seed, and number two was the color of the seed. Now what we saw in the shape of the seed is there are two variants in the shape of the seed, that is round, controlled or expressed by a factor or an allele, capital R, and wrinkled shape, which is controlled by a factor or an allele, which is small r. Likewise, for the color of the seed, one color that was considered was yellow, which was controlled by a factor or an allele represented by capital Y, and the other, green, which was controlled by a factor or an allele, small y. So here we see we are taking two pairs of characteristics. One is the shape of the seed and the other one is the color of the seed. So according to the law of independent assortment, it means that during gamete formation, by the F1 hybrids, please refer to the previous explanation that was given to understand this F1 hybrid. This F1 hybrid carrying two pairs of contrasting characters will assort independently. Meaning, if I consider round shape of the seed Then during the gamete formation by F1 hybrid, the factor controlling round shape that is the factor capital R is independent of yellow or green color. Yellow or green color controlling factors to get into the gametes. This means the factor that controls round shape of the seed can combine with yellow color controlling factor or green color controlling factor when the gametes are formed. So finally, we conclude the law of independent assortment, and here we have tried to understand that out of two pairs of contrasting character, the example that is taken out here is round shape, wrinkle shaped, yellow color, green color. A pair of factors will be independent to assort with respect with the other factors. And it is very simply explained where it has been stated that if we are considering round shape controlling factor is allowed to enter into the gamete, then the round shape controlling factor will enter into the gamete either with yellow color controlling factor or green color controlling factor. Thus, round shape controlling factor is independent of yellow color or green color. So this brings us to the end of Mendelian genetics as far as class 10 is concerned. Thank you, and I hope to meet you in another session with some other important topics related with genetics. Till then, stay home, stay safe, stay healthy. Thank you.